Okay, we are going to get started. So, my name is Dr. Devon, and today we're going to be talking about immunity mediated by B cells and antibodies. Now, before we get started, I'm going to give you a little bit of background about myself, because you might be asking yourself, who is this guy and how do we know he knows anything? So right now I'm a postdoctoral fellow. I did my undergraduate degree at CSU Chico in biochemistry. I graduated in 2012, and from there I went straight to grad school at Texas A&M, where I got my PhD in medical science in an immunology lab that studied B cells. My dissertation was based on developing a new mouse model of the central nervous system effects of Lyme disease. And that's actually what I'm currently focusing on, is I'm extending that model in Dr. Brissett's lab here at UND. And so we're looking at the effects of acute infection on the central nervous system and what the consequences of immune responses are to pathogen. So in terms of other experience, I've worked for 2 biotech startups. One of them used qPCR-based diagnostics for different fungal infections. And then I also worked for another startup where we were using therapeutic peptides to try and come up with a new treatment for sepsis. So I've done a few things. Now, getting back to the topic at hand, we're going to be today talking specifically about the development of effective antibody responses. And so where in the body might you find antibodies? And the answer is pretty much everywhere, right? But specifically there are 4 locations. So we're going to find antibodies in the blood, in the lymph, extracellular fluids, and in mucosal surfaces. Now the type of antibody in terms of the isotype that we find in these different places might not always be the same, but we'll get into that in the next lecture. Now, I think that most of you probably already know this, but I would be remiss if I didn't point it out explicitly. But antibodies are secreted by B cells and B cells only. Nobody else produces antibodies. And they have 3 main functions, and this is very important. So they neutralize pathogens or toxins, and so by neutralize I mean they surround the protein of interest with antibody, and they prevent that protein from binding any receptors that might cause pathology. They also opsonize different bacteria or proteins for phagocytosis. And so in doing so, they basically mark or tag them for phagocytosis, mostly by macrophages. And they also facilitate some of the reactions necessary for activation of complement, which is something that you've learned about already. So I think that if we're going to be talking about B cells and how they mature and how they develop this initial antibody response, we need to think about where it happens and put this into a anatomical context. So here on the left, I have a diagram of a lymph node, and you'll find this diagram in one of the assigned articles that I put up. And so let's first talk about fluid dynamics. How do things come in and out of the lymph node? So we have afferent lymph, which is the input for lymphatic fluid. This afferent lymph is first going to go into the subcapsular sinus. Now the floor of the subcapsular sinus here is not that permeable, so things sort of have to slowly trickle through these conduits, and these conduits give lymph the ability to get into the follicles where FDCs can pick up any antigen or antigen complexes, go through T cell zones, and eventually get down and trickle through into the medullary sinus, which is also called the medullary cord. Those are the same things. And in the end, they will exit through the efferent lymphatics. Now, if you're trying to keep afferent and efferent sorted out and remember which is which, just remember efferent starts with E, and the word exit also starts with E. And so efferent always exits. Now, you'll notice that there's something here called the HEV. That's the high endothelial venule. And this is the region where lymphocytes from the blood are going to see cytokine or actually chemokine signals. And those chemokine signals are going to help those lymphocytes get into the T cell and B cell zones and survey antigens in the lymph and in the primary follicles, and then eventually those are going to exit through the efferent lymphatics too. So we have an input from the lymphatic system, and we have an input from blood at the level of the HEV. The output is the efferent lymphatics. Now, in terms of our cellular compartments, I've tried to color code this to make this as easy to understand as possible. So we have B cells. So we have our resident B cells, which are going to be spending time in these primary follicles here, the yellow places. We also have circulating B cells that will come in through the high endothelial venules and find their way into the primary follicles, and those can sample and leave as well. We have follicular dendritic cells. These dendritic cells are going to permanently be spending time in these follicles. We have CD4 T cells, which are going to be surveying things, but they're primarily going to be in the purple regions, which are called the T cell zones. We have subcapsular macrophages. So these live in the subcapsular space, and I'll go into a little bit more detail on those in the next slide. And then we have medullary sinus macrophages. We're not going to get too much into these, but I can tell you that medullary sinus macrophages act like you would expect macrophages to act. They phagocytose things, and they break down antigen and process it and present it. Subcapsular macrophages don't behave the way that you would necessarily expect a macrophage to behave. So let's talk about those. So if we look at subcapsular macrophages that live in the subcapsular space, what they actually do is they take antigen that's circulating from the afferent lymph, and so this red here, these are all antigen, and they take antigen and they just kind of hold on to it. They don't internalize it, they don't break it down. And these subcapsular macrophages actually go in and they transfer that antigen onto the surface of follicular dendritic cells. Now we know that follicular dendritic cells are not the same as other dendritic cells that we would talk about, in the sense that they also do not phagocytose or break down antigen. They hang on to antigen and immune complexes, and they display those on their surface so that B cells can survey them and try to bind them. So we have our follicular naive B cells. They live in the primary node. And then we have our circulating naive B cells, which, as we said, are going to come in through the high endothelial venule and sample things. And so the cytokines that are going to be responsible for, or the chemokines are going to be responsible for drawing in B cells and T cells from the blood are CCL21 and CCL19. These get cells into the T cell zone. And now if you also express CXCL13, these will draw B cells specifically into these primary follicles. And so here in this lower diagram, we have an example of follicular dendritic cells that have had antigen transferred onto their surface by subcapsular macrophages. So there are two conflicting priorities for B cell responses. You can have a really fast, low affinity response, or you can have a delayed response with really high quality and high affinity and specificity for a pathogen. So it turns out that B cells take advantage of both of these strategies, and we're going to talk about how they do that and what the timing of that looks like. So first, we're going to talk about fast, low affinity responses. And what I want you to remember is that speed equals IgM. And where this happens is in the lymph node follicle. And so here I've drawn a little arrow into where in the lymph node we would expect this to happen. So it's just this yellow region with the follicular dendritic cells. And the first step is going to involve B cell receptor binding to antigen and surface clustering of this B cell receptor. So here we have an example of cell surface IgM being expressed, and that's binding its antigen. We get clustering, and we get a series of phosphorylation events that happen at the intracellular level, and these are going to end up amplifying signal and leading to different transcription of different important genes. So step one is necessary for activation of B cells, but it's not sufficient. And this is sort of a fail-safe, because if you have accidental or low affinity binding of antigen, you want to make sure that there's another checkpoint that you go through before you end up having some kind of autoimmune or deleterious response. So So step 2 is co-receptor activation. So we have a series of 3 co-receptors that are expressed on the B cell surface: CD81, CD19, and CR2, which is complement receptor 2. Now, if you're ever reading the modern literature, generally CR2 is called CD21. And this is something that if you stay in immunology, you'll find out pretty quickly that there are multiple names for the same things. And so you end up sort of becoming a linguist in order to really understand what's going on. So here's how this co-receptor activation clustering can work. Okay, there are probably other ways where this happens. This is a little bit oversimplified, but I think it's sufficient to demonstrate our point. And so we have a pathogen here. This pathogen has been tagged by complement in the blood, C3b. Now the CR1 receptor, which is on the surface of the B cell, is going to combine with factor 1, and it's going to catalyze the breakdown of C3b into IC3b, which eventually is going to become C3d. Now C3d is the ligand for CR2, and once CR2 binds C3d in conjunction with the other proteins, CD19 and CD81, we get another signaling cascade. So what happens here? We have step 1, B cell receptor binding and clustering. Step 2, co-receptor activation and clustering. Once you have these 2 steps happening, then you've reached a threshold where you get 1,000 to 10,000 fold amplification of your signal. Now in this figure, what I want you to appreciate is that this antigen can be expressed on the surface of a bacterial cell, or it could be a secreted antigen. It could be soluble. It could be a toxin. So here's what it looks like if it's a soluble antigen. Regardless, if you get both of these signals happening, then you get activation of the B cell. So what happens when that B cell is activated? If it gets activated, you get downregulation of a molecule called SIP, and you get upregulation of CD69. The combined result of this is that the B cell stays put. It stays in this primary follicle in the lymph node where it encountered the antigen. If the naive B cell doesn't get step 1 and step 2 of signaling, SIP becomes upregulated, there's no CD69 expression, and the B cell goes on its happy way. It goes back into the blood and circulates. So there are 2 different ways that we know of that B cells can become activated. They can become activated in the classical sense that most people talk about, which is with the requirement of help from CD4 T cells, specifically follicular helper cells, and that process is a T-dependent process. These B cells are called B2 B cells, and it's what most people talk about when they're talking about B cell responses. So B2 B cells, which receive help from T follicular helper cells, can produce IgM or IgG or IgA or IgE, and it happens in this order, and there's no going back. So once you do the you cut out the regions of the exons to produce IgA, those regions are gone, and you can never go back to producing IgE and IgG in that B cell. So these are irreversible steps. Now, it's also possible for a B cell to become activated without help from a CD4 T cell. This is called a T-independent response, and typically we call these B1 B cells. Now, you do have a series of B1 B cells that are expressed during development. I think they come from the fetal liver, and I know that mice have B1A and B1B subsets, and it can get a little bit hairy talking about these because it's not clear whether or not humans also express these subsets of B1, so we're not going to get into it, but I want you to be aware that it gets more complicated than just B1, B2. There are many subsets. But B1 B cells are mostly going to produce IgM, and they're going to stop at that point. There are exceptions. For example, in Lyme disease, you can get T-independent IgG responses. Now, how exactly that happens, I'm not sure that is known yet, but just be aware that anytime I tell you pretty much any rule in immunology, within a year or two, someone's going to come up with an exception to that rule. So keep this all as a generalization and know that the the specifics are going to depend on the disease in particular. So regardless of whether you get a B1 or a B2 response, you're going to get IgM production initially, and that's going to occur within days. Okay, so this is our quick, relatively low affinity response. Or sorry, low specificity response. Now, if you look in your textbook, IgM is going to accurately be described as a pentamer, but it's drawn as a pentagon type shape. It makes sense because you would think that the geometry of something that has 5 binding sites would be a pentagon. But research that came out this year, very recently, I think it was like a month or two ago, did some structural analysis, and they found out that even though it has 5 binding sites, it's much closer to a hexagonal type shape. In fact, it kind of looks like a snowflake. So if you look at, this is an electron micrograph, this is what the actual molecule of secreted, not cell surface, secreted IgM looks like. So we have our variable regions extending out here. We have our FC regions on the inside, and you can see that there's a little linker region that leaves a little bit of space here, and it's for a molecule whose name I can't remember, but that molecule determines the activity of macrophages in phagocytosing things. So one could imagine, I wouldn't be surprised if in the near future we find that this molecule plays a critical role in whether or not a pathogen is killed by IgM. So now let's talk about the quality of antibody responses. There are 2 mechanisms that activated B cells undergo to improve the quality of their antibodies. One is isotype switching, which we sort of discussed, the conversion of IgM to IgG to IgA to IgE, or somatic hypermutation. Now, both of these processes, in general, depend on help from CD4 positive T follicular helper cells. Now remember, in general, only B2 B cells are going to undergo the processes that we are going to describe, that I am going to describe in the next few slides. So how does the process of getting T follicular helper cell help look like? So we have lymphocytes from the blood coming in through the high endothelial venules. Those are drawn in by CCL19 and CCL21. The T cells remain in the T cell zone, which is this blue area, and they interact with dendritic cells, your typical myeloid dendritic cells, that are going to be presenting antigen to them. And that's how the T cells, naive T cells, become activated. The B cells are going to be drawn into the primary follicle by CXCL13, and those are going to survey the surface of follicular dendritic cells for intact antigen or immune complexes. Now in both cases, here the BCR is binding the antigens, here the TCR is binding the antigens. These both become activated. Cytokine signaling is going to result in these going to the boundary zone where the T cell and B cell zones meet up. And here, this is where B cells are going to survey for a cognate interaction for their TCR. So here we have MHC class 2 presented on the surface of the B cell that has internalized the antigen or immune complex that's seen on the FDC, and now the T cell is going to be looking at that antigen, at that epitope presented on MHC class 2. If there's strong binding, then we have a match made in heaven, right? These two become soulmates, as Dr. Slutton would say, and they are going to migrate and do some other things. So what do those next steps look like? So at the cell surface level, in terms of the protein-protein interactions, it's not enough to have TCR and MHC binding. You need more. So you have MHC and TCR binding, then you get CD40 and CD40 ligand binding. Once that happens between the B and the follicular helper T cell, then you get activation of a transcription factor called NF-kappa B. This is going to result in the upregulation of ICAM and LFA, which bind each other. So LFA is expressed on the T cell, ICAM is expressed on the B cell. The result of this is that you get cytoskeletal reorganization, and the Golgi in the T cell is going to move closer to that site of binding with the B cell, and you get back and forth cytokine signaling that helps activate both of these for the most effective optimal response. So once this has happened, these B T cell cognate pairs are going to travel down into the medullary sinus, which you see in this figure. It's called the sinus, and this is called the cord. They're interchangeable terms. What they're going to do is form a primary focus. Now, the primary focus is going to give rise to some plasma cells. Not all of the cells will become plasma cells, but some of them will. And those cells that become plasma cells are going to become IgM-producing plasma cells. Now, it's typically IL-5 and IL-6 that are going to drive this. And so that's going to result in your immediate high titer of IgM in response to an infection. But notice that I said some. What happens to the ones that didn't become plasma cells? They travel back into the primary follicle, and they start dividing and replicating to form a secondary focus. This secondary focus is called a germinal center. Now, within the germinal center, this is where somatic hypermutation and isotype switching occur. So those B cells that form the germinal center are going to become centroblasts. Those are going to undergo proliferation, hypermutation, and isotype switching. There are a lot of different cytokines and cell-cell interactions that mediate this process. The follicular dendritic cell plays a role in the production of IL-6, 15, and a cytokine called BAFF. CD40 ligand interactions are critical. If you don't have CD40 ligand, you're not going to get isotype switching. What does that mean? That means that you're going to have IgM only. You're not likely to produce anything other than IgM. The cytokines produced by T follicular helper cells are going to influence which type of isotype you switch to. So CD40 ligand makes it possible, and the cytokines determine how you're going to switch. Are you going to switch to IgG or IgA or IgE? So this process of both somatic hypermutation and isotype switching is mediated by an enzyme called AID. And I thought I had included the what that stood for. I think it's, I'll have to look it up. It's some kind of cytidine deaminase. Activation-induced cytidine deaminase is what it's called, and you don't have to remember that. But the point is that AID is the critical enzyme for inducing these two processes. So somatic hypermutation refers to random mutations in the variable region of the B cell receptor. That's the part that binds antigen. So these mutations are going to affect the affinity for antigen. They might increase affinity, they might decrease affinity. Now, isotype switching is going to be mediated, again, by AID, but in this case, it's going to be deleting exons within the FC region locus. And so once those are excised and pulled out, you can never get them back. Now, these mutations are going to affect which type of cell can respond to that antibody, who can see the FC region of that antibody to exert the next step of the response. Because as we know, maybe I didn't mention this, but antibodies are not inherently toxic. They're just a way of marking things for the immune system to respond to in one way or another. So there's two checkpoints for these mutations. So these centroblasts that have undergone somatic hypermutation and isotype switching re-upregulate expression of B cell receptor on their surface, and they go back to the follicular dendritic cells in the primary follicle. So I've put a red square around where these interactions are happening. Now, because the V regions of these antibodies were mutated, those mutations could result in a weak binding of the BCR to the antigen or in a strong binding. So if those mutations were not helpful and we get weak binding, then those B cells, those centrocytes, they die by apoptosis. They do not receive survival signals. However, if there's a high affinity interaction, you get clustering, you get intracellular signaling, and ultimately you get a survival signal going from the follicular dendritic cell to that centrocyte. Then you move on to the next checkpoint. So the first checkpoint checked for strong interactions between the B-cell receptor and the intact antigen presented on the follicular dendritic cell. The second checkpoint checks the ability of that centrocyte to internalize that antigen, process it, and then present it on MHC2 in a way that the T-cell receptor can see it. And so this checkpoint would have to occur in the presence of T-cells. So we get our centrocytes moving to that boundary zone, and now they're presenting this specific region of the antigen that we care about, which is called the epitope. They're presenting that to the T follicular helper cells. If we get strong binding between the TCR of the T follicular helper cell and that epitope presented on MHC class 2, and we get CD40 and CD40 ligand binding, then a high affinity interaction is going to result in a survival signal for that centrocyte. A low affinity signal is going to result in apoptosis. So now let's say we have the centrocyte that's gone through both of these checkpoints. It's able to see intact antigen, it's able to process that antigen and present it in a way to CD4 T cell that the CD4 T cell can see it. So now there are two cytokines that are going to determine the fate of this centrocyte. If we have a lot of IL-10, and we're talking about the earlier responses, so weeks, right, to infection, these centrocytes are going to become plasma cells. And what that means is they're going to massively increase their ability to produce and secrete antibody. Now, depending on the type of class isotype switching that has gone on, they may produce IgG, IgA, or IgE. Now, they could migrate to the medullary cords of the lymph node where they secrete antibody, and in this case, they'll be short-lived. If they migrate to the bone marrow, they'll be longer-lived, and they'll continue to secrete these antibodies into the blood or mucosal surfaces and provide a longer duration of protection or response to whatever pathogen may be invading. Now, if we're in the later responses, say months out to an infection, then IL-4 is going to drive the differentiation of these centrocytes into memory B cells. Now, what these memory B cells do is they kind of just sit on the fence and wait. And in this case, they migrate to a region called the subcapsular proliferative foci in the lymph node. This is a very recently discovered microstructure, and this is just superficial to the primary follicle. And what they do is they sit there and they wait. And if you ever get reinfected by the same pathogen, the antigens from that are going to get surveyed by these memory B cells, and they're going to instantly develop into plasma cells, and they're going to start secreting so much antibody that that infection is not going to get a chance to take hold. So here we just have a graphical description of what I just told you about, right? So we have a centrocyte is binding a T follicular helper cell. It's presenting that specific epitope of that antigen on MHC class 2. The TCR is seeing that. We have CD40 ligand interactions. We have clustering of the B cell receptor binding to the intact antigen. And here we have IL-10 production by the T follicular helper cell, and it's telling that B cell become a plasma cell. In this case on the right, we have the same scenario, except the T cell is producing IL-4, and it's telling that B cell, don't start secreting antibody yet, become a memory B cell, and do it later if you see that antigen. So here's the summary and what I want you to take away from this lecture. We know that B cells produce antibodies, and we know that there are two types of ways that B cells can respond. They can respond in a T-independent manner or a T-dependent manner. But regardless of which of those ways B cells respond, they can make IgM. And IgM selects for rapid, low affinity responses. Now, we also know that if you're a T-dependent B cell and you receive help from a T follicular helper cell, you can undergo isotype switching and somatic hypermutation. And collectively, both of these are going to increase the quality of the antibody response. Those processes take a little bit longer to happen than the production of IgM. IgM can happen within days. The other isotypes take weeks. Okay. Now, collectively, what we end up getting is a fast, low affinity response or a slightly delayed high affinity response. Regardless of which of those responses you get, the antibodies produced by those are going to play a role in opsonization, neutralization, and complement activation. In the next lecture, we're going to focus on specifically how do these antibodies perform their effector functions, and what are the different circumstances that can determine a successful versus a unsuccessful immune response. If you guys have any questions, please bring them to class on Monday. If you're just way too excited about this and you can't wait until Monday, you shoot me an email. My email is at the bottom there, and we can chat about that. So that concludes our lecture, and I will see you guys next time.